Hi, everyone. Welcome to this presentation that begins our study of animal diversity. We'll first be placing animals within the broader evolutionary history of life. Instead of just listing the phyla of animals, we're going to go through and build the animal tree step by asking repeatedly, what new biological trait evolved at this point in the lineage, and how did that trait shape everything that came after it? As we move through this material, I want you to think of animal diversity as a branching tree that grows as new developmental and structural innovations appear. Each major split in that tree corresponds to a new biological solution to a problem. Over time, those solutions have accumulated, leading to the extraordinary diversity of animal forms we see here today. Before we dive in, let's take a moment to look at exactly what we will accomplish by the end of this presentation. You will be able to list the features that distinguish the kingdom Animalia from other kingdoms. You will explain the process of animal reproduction and embryonic development. You will describe the roles that Hox genes play in development. You will explain the differences in animal body plans that support basic animal classification. You will compare and contrast the embryonic development of protostomes and deuterostomes. You will interpret the metazoan phylogenetic tree. You will describe the features that characterize the earliest animals and approximately when they appeared on Earth. And finally, you will explain the significance of the eras and periods for animal evolution and the changes in animal diversity that took place during those times. For a long time, biologists organized all life into four traditional kingdoms inside the domain of eukarya: the Animalia, Plantae, Fungi, and everything else lumped together as protists. That simple picture worked for textbooks, but modern phylogenomic studies have shown that it does not actually reflect true evolutionary relationships. Instead, we now recognize supergroups within the eukaryotes. These supergroups sit above the kingdoms and are based on shared ancestry revealed by molecular data. Animals belong to the supergroup Amorphiae. Within Amorphiae, animals fall into a clade called Opistheconta. The reason this matters is evolution. Animals share a more recent common ancestor with fungi than they do with plants. That means when we trace the animal lineage backward, we do not arrive at something plant like. Instead, we arrive at something more closely related to certain protists and fungi. This is our first major branch in the animal story. Before animals existed, there were unicellular and colonial protists within Opistheconta. No true multicellular life forms yet. That is where our lineage begins. The term Opistheconta refers to organisms whose ancestral cells possessed a single posterior flagellum. And you can see this feature in animal sperm cells today, but also you can see it here in the image in the bottom right in this what we call a flagellate. Sorry, choanoflagellates are single-celled or colonial protists, meaning they live in groups together, but not as a multicellular single organism. And they are believed to most closely resemble the common ancestor of animals. Each choanoflagellate cell has a flagellum surrounded by a collar of microvilli. It's kind of hard to see in this image here because microvilli are so small, but it's these little prongs that stick up at the edge of this collar here. The beating of the flagellum generates water flow, and bacteria get trapped against the collar and those microvilli, and they kind of get pulled inward into that collar and ingested. This feeding mechanism is strikingly similar to the feeding cells of the animals we call sponges. The feeding cells of those sponges are called choanocytes. And that similarity is not accidental, but suggests shared ancestry. Now that we understand the broader eukaryotic context, let's zoom in on the kingdom Animalia itself. The most basal animals belong to four monophyletic clades that branch off very early in the animal tree. These are Porifera, the sponges, Ctenophora, the comb jellies, Placozoa, small animals resembling amoeba that look almost like living blobs, and Cnidaria, which includes jellyfish and their relatives. All four of these groups lack bilateral symmetry. The remaining animals belong to the large clade called Bilateria, which contains everything else and is defined by bilateral symmetry. All animals are chemoheterotrophs, which means they obtain their energy and organic carbon through consuming other organisms. They display a wide range of feeding strategies, including carnivory, herbivory, omnivory, and parasitism. Almost all animals possess specialized tissues organized for efficient function. The pressure to acquire food has driven the evolution of motility in most species of animals, even if that movement occurs only during certain life stages. Finally, animal development follows a defined path that establishes a predetermined and fixed body plan during the embryonic stages. Animal tissues evolved to solve specific problems. Food acquisition required structures for capturing and processing prey. That led to digestive systems and, in many lineages, complete digestive tracts with separate openings for ingestion and elimination. Sensory structures evolved to detect environmental cues such as light, chemicals, and mechanical stimuli. Neural communication allowed rapid coordination of muscle contraction and behavioral responses. Intercellular communication systems allowed cells to specialize and coordinate activities. The evolution of tissues and organ systems made animals more efficient competitors in ecological systems. Each of these innovations increases structural complexity, and as we move up the animal tree, we will see how those complexities accumulate. Let's begin with the development of animal tissue. True animal tissues consist of groups of similar cells that perform specialized functions. Unlike plant cells, animal cells lack cell walls, which allow flexibility in body structure and movement. Cells are embedded in an extracellular matrix that provides support and communication. And examples of tissues include epithelial tissue, which which are tissues that line surfaces and forms protective barriers. Connective tissue, such as bone and cartilage, muscle tissue for contraction, and nervous tissue for signal transmission. The emergence of organized tissues is a defining trait of the group called eumetazoa. Sponges lack true tissue, although they have specialized cells. The difference becomes the first major evolutionary split within Animalia. We are now ready to formalize that split. However, first, let's do an assessment question to gauge your understanding of the material so far. Checkpoint question 1 asks, which of the following is not an example of epithelial tissue? To the right, you will observe our taxonomic tree of Animalia. At the top is the clade porifera, and highlighted in the green box is everything else, the eumetazoa. Eu is derived from the Greek word for true, meta for after, and zoa for animal. In other words, what scientists are trying to describe is eumetazoa means a well developed animal. What scientists mean by this is that all members of this group have organized complex tissues and go through a process we'll describe in the coming slides called gastrulation. And that's what we usually associate when even just day-to-day observations conclude, oh, is this an animal or is this not an animal? While porifera are considered animals, they're not considered to be the quote unquote true animals or eumetazoa. So this group excludes porifera, the sponges, and because porifera is next to the eumetazoa, in a taxonomic tree, you'll often see it referred to as the parazoa, as I have written in here. And para just means beside or next to. When we look at animal reproduction and development at its most base form here, animals are all diploid organisms. That means they have two copies of each of their chromosomes throughout their life with diploid somatic cells. And somatic just means cells that make up the body. These aren't germline cells, which have to do with cells, reproductive cells for generating offspring, but just cells of the body. They're all diploid and they have haploid gametes. And haploid gametes, they have half the chromosomal number of the somatic cells. That's where the term haploid comes from. And gametes are cells that are responsible for sexual reproduction. And they're generated through a process we call meiosis. This is different than most of animals' close relatives. For example, the fungi, they live most of their lives out as haploid cells. So it's a major difference in how animals go through their reproduction cycle. However, there are some exceptions to this rule, and they're worth pointing out here. For example, male hymenopterans, this includes bees, wasps, and ants, develop from unfertilized eggs, and they'll remain haploid for their entire lives. Most animals reproduce sexually, yet some groups can also reproduce asexually. In asexual reproduction, a portion of the parent's body separates and grows into a new individual through budding or fragmentation. These two modes allow animals to balance rapid population growth with genetic diversity. And with that, we're going to now transition from asexual to sexual reproduction, which leads directly into the embryonic stages we'll examine next, where the zygote begins the process of building the adult body plan. In sexual reproduction, the zygote is the beginning of every animal's development. The zygote forms when two haploid gametes fuse together, restoring the diploid form of the somatic cells. From that one cell, an entire organism must be created, and therefore the body has to be organized in order for the individual to develop. The key point here, though, is that development is not random. It's a highly regulated process in which position, timing, and cell identity are precisely controlled. Early in development, primary germ layers are established. And when we say germ layers, we're not talking about naughty microbes that cause disease. Germ layers is just the name that scientists give to these early embryonic layers of cells. These germ layers are the foundational tissues of the embryo. They are not organs themselves. Instead, they are developmental layers that will later give rise to specific tissues and organ systems. In animals, the three possible germ layers, starting with the outer layer first, are the ectoderm, endoderm, and mesoderm. As development proceeds, cells within each layer differentiate and specialize. That specialization leads to tissues, then organs, and eventually organ systems. Therefore, all tissues can be categorized based on which initial germ layer they are derived from. This progression from germ layer to tissues to organs is essential for understanding both anatomy and evolutionary relationships. As we move forward, notice that germ layer number will become one of the major branching points in the animal phylogeny. This reaches back to our first lecture on evolution, when we established that developmental biology is not separate from taxonomy. Rather, it's an artifact of our evolutionary lineage. The formation of these layers sets the stage for a process called gastrulation, the next major step that creates the primitive gut and establishes the body's basic architecture. So you'll see how this sequence of events plays out and explains why every animal ends up with a particular or fixed body plan that's determined in this early development. Okay, so that was a lot for you to take in there all at once there. So let's go through this stepwise, all right, with the blastula development. After zygote formation, which is the very first cell you see here, development directly proceeds with cleavage, which is a series of rapid mitotic divisions of the zygote. So it's not meiosis taking place here, it's just simple mitosis where the cells are dividing. However, cleavage differs from typical somatic cell division because there's no overall growth during these early stages. And instead, the embryo is subdividing the original cytoplasm into smaller and smaller cells without increasing total volume. So you notice the zygote and the 8-stage cell, there's not as much of a difference in size there. It's just more about separating the cells so then we can begin our specialization process. After 3 rounds of division, the embryo reaches the 8-cell stage. Continued cleavage produces a solid ball of cells called the morula. As divisions continue and fluid accumulates inside, the embryo becomes a hollow sphere known as the blastula. Once established, let me back up here. Once established, the blastula creates the basic cellular arrangement, and this allows for the next major development event that we talked about on the last slide called gastrulation. But at this stage, cells are still relatively similar, but the dramatic diversification of cells is going to occur next. Okay, so the cells of the blastula continue dividing, and the cells rearrange into the process of gastrulation. Cells of the blastula fold inward in gastrulation. If you look at the conversion of the blastula to the gastrula here, you'll note a digestive cavity forming on the inside. Gastrulation is what it kind of sounds like, the formation of a primitive gut. And this is called the archentrion, which is essentially a digestive cavity. Depending on which layer, the cells of germ layers become programmed to develop into varying tissues or particular organs and organ systems. And this is called organogenesis. In diploblastic cells, 2 layers form. In tripoblastic animals, a 3rd layer called the mesoderm forms between the ectoderm and the endoderm. Let's look at the characterizations here based upon embryo development. Most animals separate tissues into germ layers during embryonic development, and this allows us to categorize animals as diploblastic or triploblastic. Diploblasts possess 2 germ layers, an ectoderm and endoderm. Between them is a gel-like layer that is called mesoglia. This layer, it's not a true mesoderm. Animals such as, and we'll talk about them, but I'm just going to list them here, the cnidarians and ctenophores fall into this category. Triploblasts possess three germ layers: the ectoderm, mesoderm instead of the mesoglia, and the endoderm. The addition of mesoderm allows for greater structural complexity in triploblasts. It allows the formation of muscle tissue, complex organ systems, and more advanced body architecture becomes possible. Again, each germ layer has specific developmental fates. The endoderm forms the inner lining of the digestive and respiratory systems and gives rise to organs such as the liver and pancreas. The ectoderm forms the outer epithelial covering and the central nervous system. And then you should be aware that the mesoderm forms muscle tissue, connective tissue, and most other internal organs. The presence of mesoderm is transformative, as you could see. It allows for true muscle development, complex circulatory systems, and internal body cavities. Without mesoderm, structural complexity is going to be limited. Diploblastic animals often possess multifunctional cells. For example, epitheliomuscular cells can serve both covering and contractile functions. In contrast, triploblastic animals can compartmentalize functions into distinct tissue types. Okay, at this point, we can clearly articulate the first major split in the animal lineage. Parazoa, represented by sponges, lack true tissues and do not undergo gastrulation in the same organized way as the other animals. Oop. Eumetazoa possess true tissues and undergo gastrulation. All diploblastic and triploblastic animals fall within the eumetazoa. This means that the evolution of organized tissues and germ layers marks the foundation of most animal diversity. So now we know that the eumetazoa have the ability to form complex tissues, but they needed a way to also organize their shape into an array of different plants. And to do this will require another major step in evolution. But first, let's have another assessment question. Checkpoint 2. It asks, what key difference distinguishes eumetazoa from porifera? One of the observations biologists have made was that many animals of varying degrees of body complexities had a shared morphology during early developmental stages. Interesting, right? Lo and behold, the discovery of homeotic genes was made to help explain why this was the case. These genes determined and controlled the formation of major body parts and systems. Homeotic genes were a major leap forward in the development of complex body plans of eukaryotes, specifically what we call the bilateria, which function to control major body plans. So, for example, they control the formation of such things as legs, antennae, wings, etc. Where they've put them, where do they form? So do we put antennae at the head or the thorax of an insect? Legs on the abdomen or the head? How many segments of the body there are going to be? And so on and so forth. They do so in a manner that organizes parts of the body from the head, called the anterior position, to the tail, called the posterior position. Let's dig in a little bit more here on homeotic genes and describe some of their traits. First, they're highly conserved. That means that the genes mutate slowly. In other words, they have only slow changes to the genes and therefore also the traits over time. So each nucleotide, which is the genetic letters, they tend to resist change over time. This is because small mutational changes are often lethal or catastrophic to the function of the organism. Just a single mutation in a Hox gene could result in an extra body part, a new set of wings or lack thereof, for example, or it could cause them to occur at the wrong location. Occasionally, such changes can be beneficial and mark important steps in animal evolution. These Hox genes are also called homologous, meaning they are inherited genes sharing a common heritage. And the genetic sequences are highly similar. Um, so, across a multitude of species, we see a little change, and that means that they're going to be very similar in their genetic sequence. In fact, in all eumetazoa, they're very similar. And while these homeotic genes are shared across major groups, leading to similar embryonic structures, they are often overridden later in development so that adult forms can take their proper arrangement. Checkpoint question 3 asks, which germ layer is responsible for the formation of the following structures, the muscles? Another observation of animals is in their symmetry, which is the arrangement of the body where one or more plane can divide the animal into two mirror images. We'll discuss some symmetry terms, then see how they fit into our taxonomic tree. We've already talked about it a little bit, so you might kind of see where we're going here. And then we can look at how we can describe the body based on its orientations and types of divisions. Animals can have bilateral symmetry, which means that there are left and right halves that relatively reflect the other. They can also have radial symmetry, meaning that anywhere along a central axis, the body can be divided in half to create two equal halves. Think of a pie or a cake where you can divide it anywhere along its central axis to make two halves that are equal. And asymmetry is, of course, where the organism cannot be divided in any way into two mirror images, A meaning without, so without symmetry. Here we can see bilateral symmetry, as in Drosophila, the fruit fly. By dividing the body lengthwise, we have a bilateral symmetry of a left and a right half. We can also see how that contrasts to radial symmetry. There is a central axis that runs through the center of the flower, and along which we can divide the flower into equal halves. Finally, we have spherical symmetry, which refers to an organism that is shaped like a sphere, and any plane cut passing through the center divides the body into two mirror images or halves. And this symmetry is not important for us here in our discussion of animals because we don't see it in animals typically. This type of symmetry is ideal for free floating organisms in an environment with no preferred direction. Those are planktonic animal or planktonic life forms. And this symmetry just allows an equal interaction from all angles. But for animals, interactions are very important. As you can imagine with the fly here, the head is at the anterior end. And so we have the fly has sensory organs there in order to interact with its world as it moves through it. And for each group of organisms, that sort of directionality and also symmetry is going to be essential. Asymmetry, on the other hand, includes sponges, and you'll see with sea sponges that they want to be able to take in things from all around them, and so they might not have a symmetrical form like other organisms would. You'll even find some organisms, such as flatworms, they're bilateral in a larval stage, but they'll end up losing it as adults. And this brings us to our next grouping in our taxonomic tree, the bilateria. The bilateria is a very large clade of eumetazoans, and they have two important characteristics. One is their namesake. They evolved bilateral symmetry. This symmetry reflects a stable anterior to posterior axis and coordinated left and right development. With bilateral symmetry comes cephalization, meaning concentration of nervous tissue and sensory tissues at the anterior head or the head end. This promotes active movement and more complex ecological interactions. The second being that they are triploblastic. Remember that the presence of a mesoderm in triploblastic animals supports this increased complexity. So at this point in our tree, we have moved from simple multicellular organisms without true tissues to animals with organized tissues to animals with three germ layers and bilateral symmetry. Each step corresponds to an increase in structural and developmental sophistication, so to speak. We can now classify animals based on symmetry. Bilateria possess bilateral symmetry, and Cnidaria are radially symmetrical. Ctenophora exhibit rotational or biradial symmetry. And this is sort of a cousin to radial symmetry, and we're going to cover this shortly. Finally, porifera are the asymmetrical ones we've discussed. By placing symmetry onto the phylogenetic tree, we can see how body plan and its organization corresponds to evolutionary branching. Asymmetry is typically seen in parazoa and placozoa. These animals lack stable developmental axes. True animals, meaning eumetazoa, can be categorized based on symmetry. Cnidarians exhibit radial or biradial symmetry, ctenophora showing rotational symmetry. Again, more on that in a moment. Bilateral symmetry characterizes the bilateria. An important nuance is that evolution selects for what works here. It doesn't care about sticking to strict morphological based taxonomic lines. So, for example, in echinoderms, these are organisms within bilateria, but adult echinoderms exhibit radial symmetry. However, their larvae are bilaterally symmetrical. Um, and so this reinforces the importance of embryology in classification. Usually is better if you're just looking at phenotypical features to study the embryology to give cues into what their true evolutionary relationships are. And that is reinforced through our genetic sequencing of these organisms as well. As we cover each of these groups here and in the future presentation, take note that symmetry types are body architectures and they don't represent a ladder of progress. Instead, they represent different solutions to ecological and developmental challenges. You see, for example, our sea sponges, they don't need bilateral symmetry. They don't need to have a cephalization where they have a concentration of nerve endings in a head because they're, for the most part, sessile. They stay put on the sea floor. So always be aware that evolution is just doing what works here, and not any one of these body plans means that it's better than another body plan. It's just a different evolutionary path. Now, with that said, let's go ahead and begin with the asymmetry of porifera. The image on this slide shows an example of a sponge, and you can see the asymmetrical nature of these animals. Sponges have no consistent plane that can divide the body into mirror images. And this lack of symmetry matches the simple organization of parazoa. They lack true tissues and gastrulation. The asymmetry reflects the sessile or non moving lifestyle of most sponges and the reliance on water flow rather than any directed movement. Placing porifera at the base of the tree using this character reinforces the early divergence that we discussed earlier with Eumetazoa. Radial symmetry is seen in the phylum Cnidaria. We can see it in examples such as jellyfish and adult sea anemones. This arrangement allows the animal to experience the environment equally from all directions. And this image here on the right shows a sea anemone, and it highlights the oral side at the top here, and that's the side that contains the mouth, and what we call the aboral side, which means the base here, that would be the side furthest from the mouth. So if you imagine a pole or an axis going from the top down to the other end, then you can divide this animal in half at any plane as long as you follow that axis all the way down. And by the way, oral aboral, this terminology is used for organisms that don't have bilateral symmetry, as opposed to the anterior posterior terminology we use for bilateral organisms. Ctenophoria displays rotational symmetry. I told you we would revisit this again. And this division of the body breaks the organism down into two halves again along the oral aboral axis to produce two copies of the same half, but it only occurs rotated at 180 degrees. In other words, the body is symmetrical along a radial axis at 90-degree intervals. So if you were to cut a piece of pie in half, you'd have it symmetrical on both ends, or you could rotate that knife another 90 degrees from that first cut. And if you were to make a cut again along that piece of pie, again, it would be symmetrical on both sides, but not other angles other than that. The image on this slide illustrates that pattern, and this type of symmetry is unique to comb jellies and further refines the placement of Ctenophora on the phylogenetic tree as a separate basal lineage from the Cnidaria. Back to our phylogenetic tree, if you look at the green star there, you see the comb jellies, Ctenophora, as a separate group from Cnidaria. This slide is going to be a bit busy, but it makes more sense to see it all together rather than separate. Here we are going to see how a bilateral body can be organized or described based on its orientations and planes of division, using a human body as an example. Bilateral symmetry involves dividing the animal through a mid-sagittal plane. And what a mid-sagittal plane is, so here's the sagittal line right here, and mid means just right down the middle. And the result should be, if it has bilateral symmetry, is two mirror images of the left and right sides. Animals with bilateral symmetry have distinct anterior and posterior ends, meaning they have a head and a tail end, dorsal and ventral surfaces. Dorsal meaning back and ventral meaning front. So think of like a dorsal fin on a fish. And the plane of division we're most familiar with generating the left and the right sides is probably the sagittal plane. This bilateral arrangement is found throughout Bilateria, except for those that secondarily evolved radial symmetry. And as stated earlier, remember that each body plan represents different solutions to ecological and developmental challenges. So, for example, the bilateral symmetry, it promotes movement and promotes cephalization, which is the development of concentration of nerves towards the head end so that it's able to better experience and understand the environment as it has a streamlined directional motion towards. Some terms will refer to the location along the body. For example, dorsal means located at or near the back, and ventral means located at or near the front. On the other hand, cranial means located at or near the head, or anterior end, while caudal means near or at the tail or posterior end. And the diagram on this slide, it labels the different body orientations and planes. So it's good to get familiar with each one of these as I go through it. The coronal plane divides dorsal and ventral parts. The sagittal plane, again, divides left and right parts. And the transverse plane divides the anterior and posterior sections. Next is medial, and this is towards the midline, and lateral, which means away from the midline. So if you look at the top here, midline is right down the middle here, and lateral is if you go off to either side. Proximal means close to the origin of a body part, and distal means further from the origin. So take the arm, for example. You could say that something is proximal to the shoulder or near the shoulder, or you might say it's distal to the shoulder. So maybe way out here, the wrist is distal to the shoulder if you were describing it. These terms allow us to describe body organization precisely and also allow us to continue building the phylogenetic tree with consistent vocabulary. Throughout this course, both in this lecture and in our labs, these terms will be quite useful to you as we're going to use them frequently. So it's a good idea to be fluent in all of them. Checkpoint question 4. What plane would divide the body of a bilaterally symmetrical animal into right and left halves? So far, we've discussed animals based on tissue organization. Parazoa, such as porifera, lack organized tissues. However, the eumetazoa possess tissues and undergo gastrulation. Now we refine our classification by incorporating embryological traits. This tree is not simply a list of groups, it's a map of shared ancestry, remember. So each node represents a common ancestor that possessed a specific developmental innovation. As we continue, we will add additional traits to this tree, particularly those related to internal body cavities. Scientists also classify animals according to the number of germ layers formed during embryonic development. The class Bilateria is a triploblastic organism group because its members develop 3 germ layers. The group known as Radiata is diploblastic and develops only 2 germ layers. Remember, their inner layer is not a true mesoderm germ layer of cells, but rather a gel-like layer we call the mesoglia. Diploblasts possess 2 germ layers, the ectoderm and endoderm. Between them is that gel layer we call the mesoglia. Again, it's not a true mesoderm. And animals such as cnidarians and ctenophores fall into this category. This diagram on this slide places those groups on the tree using the germ layer character we examined during gastrulation. We can further categorize Bilateria using additional developmental features of the body plan. One of the most informative of these is the presence or absence of an internal body cavity. The diagram on the slide shows how this new character integrates, where I put the 2 green stars. And so earlier we discussed the tissues, germ layers, and symmetry. And now we'll look at the details of the body cavity or the coelom itself as the next major innovation in animal body plans. Triploblastic animals receive one more important layer of organization through the development of an internal body cavity called the coelom. This cavity originates entirely from the mesoderm germ layer and sits between our outer body wall and the internal organs. Fluid fills the space and creates a cushion that protects delicate organs from mechanical stress during movement or external impacts. The fluid allows organs to shift and expand independently of the body wall, which supports more efficient digestion, circulation, and growth. Over evolutionary time, the coelom opened the door for larger body sizes and more active lifestyles because muscles in the body wall could contract without dragging the gut or other viscera along with them. When we add the presence or absence of the coelom to the developmental characters we have already collected, this trait becomes a clear marker that helps us draw additional branches on the animal phylogenetic tree. The diagram on this slide illustrates exactly how the coelom divides the body into functional compartments. In mammals, the cavity separates into a thoracic region. This encloses the heart and the lungs, and an abdominal region that houses the digestive organs. The fluid inside these compartments absorbs shocks and maintains a stable environment for the organs while the body moves. Because the space also allows gases and nutrients to diffuse freely between the tissues, it supports higher metabolic demands. These functional advantages explain how the evolution of a true coelom coincided with the appearance of more complex and mobile animals across multiple lineages. Placing this trait on our growing phylogenetic tree now lets us group animals according to shared body cavity architecture. This brings us to our checkpoint question 5. What is the coelom in triploblastic animals? Triploblastic animals show 3 different patterns of body cavity development that represent important evolutionary grades. In acoelomates, the region between the digestive tract and the outer body wall is solidly filled with mesoderm tissue, so there is no internal cavity at all. And that is the acoelomates. The flatworms in the phylum Platyhelminthes are the classic example of this condition, one which we'll be doing a dissection lab on in this semester. In eucoelomates, or true coelomates, the body cavity forms completely within the mesoderm and is lined on both sides by a continuous layer of epithelial tissue called mesothelium. This arrangement is found in annelids, mollusks, arthropods, echinoderms, and chordates. A third group, called pseudocoelomates, has a body cavity that is lined by mesoderm on the outer side only, while the inner side remains lined by an endoderm. Roundworms in the phylum Nematoda belong to this category. These 3 body cavity types allow us to subdivide the triploblastic branch of the phylogenetic tree with even greater precision and show how different lineages solve the problem of internal support and organ protection in distinct ways. Now, this slide provides us an example of each 3 of those groups. A is the acoelomates, and I said a great example of this was the Platyhelminthes, or the flatworms, and you can see an example of the flatworm in the upper left corner there. And if you'll take note, if I get out my pen, laser pointer here, if we started from the outside, we have the ectoderm, the mesoderm, and then the endoderm, and then the digestive cavity. So this body cavity in the acoelomates is not present, right? If we look, however, over at this one here, the eucoelomate, the true coelomates, and this includes a wide variety of different organisms: annelids, mollusks, arthropods, echinoderms, chordates, which humans belong to. We can follow from the outside in. We have the ectoderm and the mesoderm, and we have the endoderm all the way here in the middle. And here's the coelom present now. It's the green layer here. And you'll note that the coelom is entirely surrounded by the mesoderm. If we hop over to the pseudocoelomates, I explained that They have one type of germ layer on one side and a different type of germ layer on the other side. So from the outside inward, we have the ectoderm, then we have the mesoderm, and then we have the pseudocoelom. And instead of having the mesoderm on the inner portion of the coelom, instead in pseudocoeloms, you have the endoderm present. Now we'll go ahead and integrate the coelom character into the growing phylogenetic tree. It shows how the presence or absence of a body cavity separates major groups once we have already established tissues, germ layers, and symmetry. Note that note sorry, please notice on the slide that some descendants secondarily lost the body cavity, such as the flatworms we just discussed. This visual model is going to help you to see the evolutionary history, not always a simple progression of adding new features. Sometimes lineages simplify or modify earlier innovations, but the tree is beginning to take shape as we layer each new developmental character on top of the previous one. Within the coelomates, we can take the classification one step further by adding the final developmental character, the origin of the mouth and anus from the blastophore. The slide organizes animals into protostomes and the deuterostomes based on this trait and shows how it overlays with the coelom character we've already placed on the tree. So at the top here, we have the protostomia, and the near middle round here, the bottom of the coelomates, we have the deuterostomia. Now let's go ahead and cover what exactly those terms mean. Triploblastic eucoelomates can be divided into two major groups based on how the mouth and anus form during gastrulation. When the primitive gut first contacts to the outside of the embryo, the opening is called the blastopore. So this is an early development. Here is their gastrula, and the blastopore has been formed from that inward folding. In protostomes, the mouth develops directly at the blastopore, while the anus forms later at the opposite end of the gut. In deuterostomes, the mouth develops at the opposite end from the blastopore, and the anus forms at the original blastopore site. Protostomes, looking at the top here, they're going to include arthropods, mollusks, and annelids. Deuterostomes include chordates and echinoderms. This consistent difference in blastopore fate is one of the strongest characters scientists use to separate the two major branches of bilateria. So if you look at the phylogenetic tree, it plays an incredible importance here for dividing these different organisms up. It completes the set of developmental traits we began building early on from the zygote all the way to the adult body plan. And this leads into checkpoint question 6. How do the developmental patterns of protostomes and deuterostomes differ regarding the blastopore? Having examined how embryonic development establishes germ layers and body axes, let's turn our attention to the later stages of development. Many animals produce a juvenile stage called a larva that differs markedly from the adult structure, function, and diet. Metamorphosis is an abrupt change in body structure that transforms the larva into the adult form. The Greek meaning of the word metamorphosis is transforming after forming, which accurately describes the dramatic reorganization that occurs. The series of developmental stages from embryo through larva to adult remains largely the same for most members of the animal kingdom, which is another piece of evidence that unites related groups of the phylogenetic tree and shows how the fixed body plan we discussed early in the lecture can be expressed in very different ways during different life stages. Incomplete metamorphosis involves gradual changes from juvenile form to adult form, In this type of development, the young organism resembles a smaller version of the adult, but lacks fully developed reproductive structures. Grasshoppers provide a common example. Their young stages are called nymphs. These nymphs resemble adults in general body structure, but are smaller and lack mature reproductive organs and wings. As the organism grows, it undergoes a series of molts. Each molt produces a slightly more developed form. Over time, these gradual changes lead to the fully functional adult stage. Because the basic body architecture is present from early development, the transformation is progressive rather than dramatic. Complete metamorphosis involves a much more radical transformation and occurs in distinct stages: egg, larva, pupa, and adult. The embryo first develops into one of or more feeding larval stages that often look and function entirely different from the adult. The larva is specialized for growth and feeding, frequently occupying a different ecological niche than the adult to reduce competition with the same species. After the larval period, the animal enters the pupal stage, during which extensive reorganization of tissues takes place. The adult then emerges from the pupa with a completely different body suited to reproduction and dispersal. Many insects, such as butterflies, undergo this process. The caterpillar larva feeds voraciously on leaves, the pupa undergoes metamorphosis inside a chrysalis, and the winged adult focuses on finding mates and laying eggs. Echinoderms, such as sea stars, also show complete metamorphosis, with a bilateral swimming larva that transforms into the radial adult. This pattern allows for extreme specialization at different life stages, and contributes to the ecological success of these groups. This brings us to checkpoint question 7. What is the defining characteristic of complete metamorphosis? We're going to shift gears a little bit now, and now that we have a good understanding of the different characteristics of the phylogenetic tree of animals, we're going to examine the history of them. The expansion of multicellular animal life forms occurred during the Cambrian period and was like nothing seen before or since then. It began about 540 to 500 million years ago in what we call the Paleozoic era. It's referred to as an explosion due to the numerous speciation events at the time, and the oldest fossils of which are those that resemble modern sea sponges of today. Scientists debate what were the historical contingencies that caused the Cambrian explosion. One of the most important contributing catalysts was the change in Earth's atmosphere. It was about this time that we saw a massive rise in the atmosphere's concentration of oxygen. See, one of the challenges of multicellular life is the huge energy demands. Different eukaryotes were able to meet these demands in unique ways. For example, we've seen with plants that they solved this through their photoautotrophic abilities, while animals were able to do this through aerobic cellular respiration, which generates massively more ATP than simple anaerobic respiration. And in the presence of oxygen, they were able to do aerobic cellular respiration, something that wasn't really done or had evolved before the great oxygenation event on Earth. So, we're going to walk through our phylogenetic tree in the next few slides using what we've learned so far. And in the next several presentations, we'll explore each one of these groups in the phylogenetic tree in much greater detail. But this will just be a very broad, quick overview. Returning to our phylogenetic tree, We can now see how the major clades relate to one another. Eumetazoa can be divided into two broad groups. One includes animals with radial body organizations, such as the cnidarians, and the other includes Bilateria, which possesses bilateral symmetry and three germ layers. Cnidarians remain outside Bilateria, but still within Eumetazoa. Within Bilateria, developmental differences produce two major lineages that have a body cavity: protostomes and deuterostomes. The deuterostomes include the chordates, which are the animals we are probably most familiar with, such as mammals, lizards, reptiles, birds, etc., but also a group called the echinoderms, which includes also some familiar animals such as starfish. Protostomia first are broken up into two clades, the ecdysozoa and, sorry, let me back up here. I think we removed the image of the two. The ecdysozoa and the lophotrochozoa. The ecdysozoa is made up of the arthropods, such as crustaceans, beetles, and millipedes. The name comes from the process of ecdysis, the periodic shedding of the exoskeleton or cuticle that allows these animals to grow. This molting strategy is a defining feature of the clade and links roundworms and arthropods despite their very different body forms. Also belonging to the ecdysozoa are the nematodes, which covers roundworms such as the hookworms and pinworms. The final group of the protostomia is being the lophotrochozoans. Lophotrochozoa is named for two structures found in many of its members: the trochophora larvae, which is a free swimming larval stage with a band of cilia, and the lophophore is a crown like feeding organ in some adults. You put the trochophore and the lophophore together, and you get the lophotrochozoa. These shared larval and feeding structures unite groups such as the annelids, mollusks, and several smaller phyla that belong to the Lophotrochozoa. But that wraps up our discussion on animal diversity, and we'll spend the next several presentations surveying each one of these groups in more detail.